Okay, so this is chapter 12. So we just finished up cell communication and how cells communicate with one another, and they can communicate. We looked at some specific examples with the nervous system and the immune system. All right, so now we're going to look at how cells divide. And so we're going to see there's a little bit of communication with this as well towards the end. But this is called the cell cycle. This is the basic way that eukaryotic cells divide. So let's look at this. So we're going to write unicellular organisms. When the cell divides, you produce a whole new entire organism because the organism is only one cell big. But in multicellular organisms like us, we depend on cell division not necessarily to replicate, but to develop. So everybody starts out being a fertilized cell. That is when egg and sperm meet and makes a zygote, and then it divides and grows. And so that goes to the next thing, growth. We become more cells big. That's what happens when we grow, is that our matter gets bigger we become more cells big. And then our cells divide to repair. So when there's damage done, we can make new cells to repair ourselves. And so this cell division is part of a larger process called the cell cycle that we're going to talk about here. So it says the life of a cell from formation. So when a cell first forms, and all cells come from pre-existing cells. So when a cell forms, that's from another cell previously dividing. And so what happens to that cell from the time that it's formed, from that previous division, to when it itself divides? That time and that process in between those 2 points is called the cell cycle. So what happens is, for 12.1 here, is that you get genetically identical daughter cells. The daughter cells is the name for the cells that are going to come at the end. So if you have one cell, and that cell grows and divides, and now you have 2 cells, these 2 cells are called daughter cells. And so this is sometimes called the parent cell. So cells, what they have to do is duplicate their genetic material before they divide. So we have DNA in the nucleus, right? In the nucleus of every cell. And so what happens is our DNA is wrapped around proteins and are used to form something called chromosomes. And we have in our cells, in our body cells, we have 46 pieces of DNA. So let's do the math here. So if you have a cell in the nucleus with 46 pieces of DNA, and if that cell divides to be a normal human cell in the nucleus, it needs to have 46 pieces of DNA, and just not any 46 pieces. They have to have the identical 46 pieces of DNA that was in that original cell. So mathematically, to get 46 and then get 2 sets of 46, you have to make a copy of that. And so otherwise, if the cell divides, you'd only have 23 and 23, and those cells would die. And so the genetic material is duplicated, ensuring that each daughter cell receives an exact copy of the genetic material, which we know is DNA. So a dividing cell duplicates its DNA, it's real important, allocates 2 copies to opposite ends of the cell, and then splits into daughter cells. So opposite ends of the cell. So if this is a piece of DNA and it makes a copy of the DNA, what that means is the copies go on either 2 sides of the cell, so that when the cell, get a different color here, splits down the middle and you end up with 2 cells, each cell has exactly 1 copy. And so it kind of makes it organized. So let's talk about the organization of genetic material. A cell's endowment of DNA, its genetic information, is called its genome. So we have 46 pieces of DNA in our genome. DNA molecules in a cell are packaged into chromosomes. Every eukaryotic species has a characteristic number of chromosomes. in each cell nucleus. So every species may have different numbers, and so we have 46. So let me talk about DNA molecules in a cell are packaged into chromosomes. We've kind of talked about this already, but I want to review it. So there are proteins called histones, and DNA is wrapped around the histones. So these are your histones in purple. The blue is the DNA. And when a cell is not dividing, it's in a state called chromatin. This is loosely packed. When a cell starts to get ready to divide, these condense. And so during division and getting ready for division, chromatin packs tightly into chromosomes. There's a lot of chromo words in this. Chromatin, chromosomes. And so what happens is these histones are now closer together, and you get this DNA wrapped around it. So imagine a whole bunch of these. See, they're far apart and it's loosely packed. And when a cell's not going under cell division, you can't really see the DNA underneath the microscope, this chromatin. But when it's starting to go through cell division and this condenses, this chromatin condenses into chromosomes, they're short and stocky and thicker, and you can see them better underneath the microscope. So you can actually see the chromosomes in cell division. And chromosomes are only found in the cell during cell division. All right. So then we have two different kinds of cells in our body. We have what are called somatic cells. Somatic cells are non-reproductive cells. So basically, any body cell, any cell besides egg and sperm cells are your non-reproductive cells. Notice it says it has two sets of chromosomes. We have two of every type of chromosome. One set comes from mom, and one set from dad. And so that's why we have two sets. Now what happens when we go and make egg and sperm cells, those are called gametes. Those are your reproductive cells. They have half the number of chromosomes. And so they only have one set of chromosomes. So they only have, remember, somatic cells have two sets of chromosomes. That gives us a number of 46 total. Number of chromosomes. So therefore gametes have 23 total number of chromosomes. They have half the number. And that makes sense because if you have an egg that's 23 and a sperm is going to fertilize that, it also has to have 23, so that when they fuse, when the sperm fertilizes the egg and you get your zygote has 46 chromosomes, and this is a normal number for humans. We're going to see later that if we have too many or too little chromosomes, the cell usually doesn't survive. All right, so 46 is that magic number. Eukaryotic chromosomes consist of chromatin, so I just talked about chromatin, a complex of DNA and protein that condenses during cell division, so you can see it underneath the microscope in cell division. All right, so let's look at this process. So what happens to these chromosomes when a cell is getting ready to divide? So in preparation for cell division, DNA is replicated and the chromosomes condense, just like I just talked about. Each duplicated chromosome has two sister, what we call chromatid. Another chromal word. So it's chromatin over here, condenses to form chromosomes, and then when they duplicate, they form what are called sister chromatids. And these are going to separate during cell division. I'll show you a picture of this here. So the centromere, a lot of vocab here, new vocab, is the narrow waist of the duplicated chromosome where the two chromatids are most closely attached. So that's what this next picture shows. To flip it over here. So this is supposed to be a cell before it's going to go through cell division. So notice here it says chromosome duplication, including DNA synthesis. So we have to replicate the DNA. So I'm going to go ahead and draw in, let me pick a different color here. Let's do pink. Draw in, here's the DNA. I'm just going to put a curly little structure in here. That DNA is going to be wrapped around proteins, those histone proteins, condensed to form a chromosome. And so it's already gone from chromatin, all right, to chromosome. And then that DNA is replicated. So let's draw here. What does that mean? Here's the DNA. We make an exact copy of this DNA. And so here's one copy. Here's another copy. And these copies are held together at this indentation. See how this indents here? That's called the centromere. So that's the indentation. And that's holding together the two sides. The sister chromatids have your two identical strands of DNA. So here's one identical strand, here's another identical strand. So we have two sister chromatids here. And so these are identical DNA strands in each one of those. And then the whole thing is one duplicated chromosome. Why? Because it has a copy of its DNA here, so it's a duplicated chromosome. So what's going to happen during cell division, so now we've made a copy. Remember, we know we have to make a copy because if we have a cell in humans with 46 chromosomes and it's going to divide in half and make two new cells, each new cell has to have 46 chromosomes. So it has to have one of each copy of DNA. And so here we have, it makes a copy of that DNA, and then one copy goes to one cell and one copy goes to another cell. So technically, these two copies are going to split and go to opposite sides of the cell, and that cell is going to split right down that center here, and you're going to get one copy over here and one copy over here is essentially what happens during mitosis. So let's look, that's kind of the overview. This is an actual picture of this duplicated chromosome with two sister chromatids. So the eukaryotic cell division consists of mitosis and what we call cytokinesis. Mitosis, notice there is the division of the nucleus, and then the cytoplasm is going to divide. And so I'll show you some pictures here of that. So gametes are produced by a variation of cell division called meiosis. They need their own cell division because you're going to end up with half the number of chromosomes. So meiosis yields non-identical daughter cells that have only one set of chromosomes, half as many as the parent cell, which for humans is 23 chromosomes. So remember, mitosis is going to yield two identical cells that are each going to have 46 chromosomes. All right, and we'll look at another chapter that deals totally with meiosis, but we're just introducing the fact that meiosis is a special cell division. So then let's look at what actually happens in mitosis. So the mitotic phase alternates with interphase in the cell cycle. So Walter Fleming developed dyes to observe chromosomes and what happens to them during mitosis and cytokinesis. So what goes on in the movement of these chromosomes so that you end up with two identical daughter cells at the end. So to Fleming, it appeared that the cells simply grew larger between one cell division and the next. So he thought that the cell grows, gets bigger, divides in half. And that does happen. The cell does get bigger, because if it didn't, your cells with each division would get smaller and smaller and smaller. But there's some other things that we now know that occur. So let's look at the process of the cell cycle. It consists of the mitotic phase, or sometimes called the M phase for mitosis, and cytokinesis. And then interphase, which what happens here is the cell grows, so we have cell growth and copying of the chromosomes. So key here is it's prepping for cell division. So the cell spends most of its time in that phase and little time in the mitotic phase. So if we look here at this next picture, it gives you kind of a pie chart of kind of time spent in each one of the phases. So notice here that interphase is all the way, I'm going to point here, this arrow here, all but in my little part, this yellow part, this little slice of the pie, the rest of it's interphase. The cell spends, let's write up here, about 90% of the whole cell cycle, this whole process. Remember, from when a cell is made to when it gets ready to divide is the cell cycle. And so 90% of its time is prepping for cell division. And so notice that the phase interphase has 3 subcategories, G1, S, and G2. So that's what this is. G1 stands for first gap, S stands for synthesis, and G2 is the second gap. So basically the cell is getting ready to divide. And what does the cell do when it's getting ready to divide? Going back to the last page, it grows and it copies its chromosomes. So if we see here, so G1, it grows. S, DNA synthesis, all right. This is the copies the DNA. And G2 grows some more and prepares to divide. It's in this yellow part that it actually divides. The mitotic phase consists of 2 subphases, mitosis and cytokinesis. So mitosis is divided into the 5 phases. So they're not shown here, but there are 5 little phases of mitosis, and that's prophase, prometaphase, metaphase, anaphase. Oops, I'm sorry, I didn't realize it was off the thing. So conventionally divided into 5 phases: prophase, prometaphase, metaphase, anaphase, telophase. So there's some things that go on with the chromosomes here during mitosis. Remember, mitosis is the division of the nucleus. And so during this, what's going to happen is we're going to go with from a cell with 1 nucleus to a cell with 2 nuclei. And then that cell is going to divide in half, so that's called cytokinesis, is the division of the cytoplasm. Now remember that this is only a small part of the cell cycle. So most of the time the cell is preparing to do these things, and then these things, with mitosis, with these 5 subcategories, and cytokinesis occurs very quickly. When the next page, if you look here, is blank, and I meant to have a picture that looks like this and this. I had it all on one sheet for you to look at, but for some reason I didn't notice it didn't copy. So what I did, if you want to pause this right now, I uploaded onto my website. As long as you're logged in with a clarkston.k12.mi.us address, you can access these and print them out. They're just from your book, and it's figure 12.7 in your book. I labeled them on my website as mitosis picture A and mitosis picture B. So if you want to print those out, you can. All right, so that as we go through it, you can look at them. All right, so what happens? So during mitosis, so I said that the interphase, G1, S, and G2 preparing to divide. When it's ready to divide, it's going to leave the G2 phase and enter mitosis and enter the first phase of mitosis, which is prometaphase. And that's what we see here. So this is the G2 of interphase. All right, so right here, from here on out, this is mitosis. This is the beginning of mitosis. Here is prometaphase. So, prophase. Let's look at the G2 of interphase. So notice that the chromosomes are duplicated. It says uncondensed though, so they're duplicated, but they haven't condensed into those short, stocky little structures called chromosomes yet. So when you look at a cell in the G2 of interphase underneath the microscope, you can't really see the DNA. You can see the nucleus, but you can't really see the DNA. Here's the nucleolus, the spot in where ribosomes are made. And here are your centrosomes. And your centrosomes replicate, and they have centrioles that are going to be involved in helping the chromosomes move during cell division. So if we look over here, back over here, we're in G2, so the DNA synthesis has happened. So that's what that means, the duplicated chromosomes. All right, so now if we look, now we enter mitosis. So now the cell has done all its prep work, has done all of interphase G1, S, and G2, is entering mitosis. The first stage of mitosis is prophase. So notice what's happening. Just look at from this picture to this picture. You can pause it and see if you can think of what happens. What happens, as you can see here, is that the chromosomes are starting to condense. We can see the two sister chromatids of one chromosome. So these guys are condensing. We're starting to see them more clearly. All right, and you can see the centromere here. That's where, remember, the two sister chromatids are attached. And notice what's happening to these centrosomes. The centrosomes are starting to separate, and then it says early mitotic spindle. This we're going to see radiating from the centrosomes are microtubules. And the aster are the small little microtubules that kind of look like stars coming off of the centrosomes. So the centrosomes is kind of like an area that contains a pair of centrioles, and the aster comes out around it. So that is prophase. So then we look over here at prometaphase. This means it happens before metaphase. Notice here, fragments of nuclear envelope. So it's breaking down. The nuclear envelope's breaking down. You can see here now your chromosomes are really condensed. They're nice and short and stocky. So that's what I mean by condensed. So very stringy here, loosely packed, more tightly packed, thicker, and shorter, stockier. You can see these underneath the microscope. Notice here the centrioles, our centrosomes, are all the way on the opposite sides of the cell. Notice here that you have these spindle fibers or microtubules that sometimes they're called. Sometimes they're called spindle fibers, radiating from the centrosomes that are now on completely opposite sides of the cell. And notice that there are two kinds of microtubules. There are the ones that come over here like these guys. See how these guys aren't attached to a chromosome, but they overlap with one another? Those are called non-kinetochore microtubules. So that's what they have here. So there's this one. They're labeling these right here, non-kinetochore. These guys are not attached to, and I'm going to abbreviate C-some for chromosome because we write it a lot. Now notice that you also have, just put them in a different color here, you also have microtubules that attach to, looks like the centrosome. There's a special spot on the centrosome called the kinetochore spot. That's why they call these the kinetochore microtubules, because they attach to that spot. And notice that you eventually, now this have a kinetochore attached to each chromosome, one from either side. So notice that this one hasn't happened, that hasn't happened yet, and this one hasn't happened, but eventually one from over here will attach right here, and one from over here will attach right here. And so you'll have one from either side. And so what that's going to do is it helps them to line up down the middle. All right, so when we look at the next phase, so next phase is metaphase. Meta means middle. Meta, you can see here that now all the replicated chromosomes, the condensed chromosomes, are lined up down the middle. This is called the metaphase plate. So that's that dotted line here. It's the imaginary dotted line, kind of like the equator. And so they're all lined up down the middle. And notice that you have each chromosome, look at each chromosome at the kinetochore has a spindle from either side attached to it. And then notice also that you also have those non-kinetochore microtubules. That's these guys here, and they overlap one another like this. All right, so this and this, this and this. Whoops, that's not supposed to attach to that one. Got a little overzealous there. All right, so these guys overlap. And so these guys have, each one of these types of microtubules have specific purposes. So if we look here, so meta means middle, that's metaphase. What happens in anaphase? Well, remember these replicated chromosomes, let's just refer, they're all lined up down the center of the cell. And so remember that one of these, each side of the sister chromatid has an exact copy of the DNA. So when we look at it drawn like this, you know, like this is a replicated chromosome. And this right here is the centromere. But remember, each, and these guys right here are your sister chromatids. And this whole thing right here is one duplicated chromosome. That each side, each sister chromatid is an exact copy of the DNA. So what happens is, if you have, in our cells, this one has 6 chromosomes. If you have 46 chromosomes like our cells, they're all lined up single-filely, and one copy is on the left and one copy is on the right attached at that centromere. So what happens is, we want each cell to have one of each one of these pieces of DNA here. So what happens is the cell, with the help of the microtubules, separate the copies of DNA. So here's one copy of the DNA, and here's the other exact copy, and those kinetochore microtubules pull those identical copies to opposite sides of the cell. So all of these guys, so this one had this copy of the DNA, and this is an exact copy of it, and each copy is being pulled to opposite sides of the cell. That ensures that one of each copy is on this side of the cell, one of each copy is on this side of the cell. So we started out, let's say, in this cell with 6 pieces of DNA. We made copies of them, and now these 6 copies are going to go to this side, these 6 copies are going to go to this side. And so that's what the kinetochore microtubules do, is help pull those copies of DNA to opposite sides of the cell. So what in the world do these non-kinetochore microtubules do? These guys help elongate the cell. Do you see from this picture to this picture that the cell looks a little bit longer? So the cell starts to elongate. And so these guys kind of push against one another and elongate the cell. That's going to ease cell division. And so then if we look at the next phase, which is telophase, and they did telophase and cytokinesis together here. This is really not cytokinesis. We'll look at the exact picture of cytokinesis in just a minute. We'll draw another picture here. This picture is actually telophase. So this is telophase. Notice here we have one cell. And so this is the end of mitosis. So this also from the previous page. So mitosis, prophase, metaphase, anaphase, telophase. This is all mitosis as well. Mitosis, remember, is the splitting of the nucleus. So from this picture to this picture, what do we have? We have each one of the identical copies on either sides of the cell. We have the nucleolus forming again and the nuclear envelope forming. So we're getting 2 nuclei forming. Each nuclei have one copy of each of the pieces of DNA. Notice here the cell is starting to indent. They call this the cleavage furrow. And so the cell is starting to indent. Eventually it'll keep on indenting and indenting and indenting, until the moment when it splits into two. So I'm going to draw here a picture here where you have two cells, each with a nucleus, two cells. That's cytokinesis, that moment where the cytoplasm is split. So that's why I said this picture is really telophase, because remember, mitosis by definition is the division of the nucleus. So look at here, we have two nuclei, but still one cell. Even though it's starting to pinch in, it's still one cell, and the cytoplasm hasn't been divided yet. And so it keeps pinching in and pinching in and pinching in, and once it does, that's cytokinesis, and you have two now identical cells. And what phase are these cells in once they divide? They are now in interphase. All right, so going back to that cell cycle picture here, here's their two cells. We just said that they just divided. Now each of these cells enter interphase, and they begin their own cell cycle until they eventually divide, going into mitosis and cytokinesis. And so that's the process of mitosis and the movement of the chromosomes. So very, very important to get it right and to get two identical cells with the correct number of chromosomes at the end. So the mitotic spindle is an apparatus of microtubules. So let's look at in detail here. So I just drew those out, and we just looked at them, that control chromosome movement during mitosis. Assembly of spindle microtubules begins in the, where do we make these microtubules? What is that spot called? It's called the centrosome. The microtubule organizing center. The centrosome replicates at the very beginning, forming two centrosomes that migrate to opposite ends of the cell as spindle microtubules grow out from them. So that's what these pictures here, it replicates, and they separate to opposite sides of the cell. The spindles are coming out here. Eventually they're on complete opposite sides of the cell with these spindle microtubules coming out. An aster, which is a little short microtubules extend from each chromosome. The spindle, sometimes they'll call the spindle, is completely these three things: centrosomes, the microtubules, and the asters, all completely are called the spindle. So aster, these guys here, centrosome, and microtubules all form the spindle. Some spindle microtubules attach to the blank of chromosomes. What's the region where they attach? It's called the kinetochores. And help them to move the chromosomes to the metaphase plate. In anaphase, the sister chromatids, what do? Anaphase, the sister chromatids separate. They move along the kinetochore microtubules towards opposite ends of the cell. So that's this part here. All right, so they're moving to opposite poles. And how the microtubules shorten, they get shorter here. So notice here, they were longer here when they were in the middle. Now they're getting shorter. They shorten by depolymerizing. That's opposite of polymerizing. Polymerize means to, polymer, remember, is long chain of many smaller molecules called monomers. So you're doing the opposite of that. So we're breaking a large molecule down. All right, so it's breaking down here. So let's look at here. What they did is they made the microtubules radioactive, and they made part of it not, and they watched what happens during anaphase as the copies of the chromosomes moved to opposite poles or sides of the cell. So these are the centrosomes. Here's the spindle, the microtubules. Some of them are attached to the chromatids, some are not. And so they're watching what happens, and you can see here that this part didn't move, but this part got shorter and shorter, which showed that the microtubules get shorter from the area where they're attached to the chromosome itself. And so this picture right here shows that. And so here's what you're looking at here is the chromatid. Here's here's where it's attached, here's the microtubule, here's that kinetochore region. And so what they found are there are these motor proteins, and each one of these, as the chromosome moves this way, this part here breaks down into smaller subunits. That's depolymerizing. And that's how they break down. Then the non-kinetochore microtubules from opposite poles overlap and push against one another. Remember, that helps to elongate the cell. And why is it important for it to elongate? Let's go back here. It gets longer because then your two copies are far apart so that when it divides, these stay on this side and these stay on this side. It's helpful in division. And then eventually telophase, genetically identical daughter nuclei form at opposite ends of the cell. And that's what we see here, opposite ends of the cell. So cytokinesis, a closer look. And so when the cell eventually splits, so after telophase and the cell eventually splits, that's called cytokinesis, the division of the cytoplasm. It looks a little bit different in plants and animal cells. So in animal cells, cytokinesis occurs by a process known as cleavage. And I showed you that cleavage furrow earlier. And I like this picture because it shows you that the animal cell is really a sphere. So this is three-dimensional. And so what happens are there are these proteins that kind of clinch and cinch the two cells and the two sides of the cell in half. So this is the cleavage furrow. So when we look at this is what's going on here. This is a cleavage furrow. There's this contractile ring of microfilaments. So they contract and kind of cinch it, like if you cinch it right in half, and then it divides into two, and you get two daughter cells. And that's what happens in animal cells, is you have this microfilaments contracting. And plant cells, remember, plant cells are a little different. They have a cell wall on the outside. So in plant cells, we have what's called a cell plate that forms during cytokinesis. And so our next picture shows this, the cell the cell plate forming. So here is so mitosis has been done. Here's your two nuclei. Now it's going to divide in half in cytokinesis to form two new plant cells. So what happens is vesicles, and this is the actual real picture of these vesicles. Vesicles formed within the cell line up down the center here, and they get bigger and bigger until eventually they fuse, forming what's called the cell plate, which eventually fuses with the current cell walls, and you end up with two daughter cells. And so that's a little bit different with plant cells versus animal cells. And this picture down here shows you, I had you guys print out the animal cell picture. This shows you prophase, prometaphase, metaphase, anaphase, and telophase for all the different plant cells. And this is an actual real picture instead of a computer-generated picture, but it tells you what exactly happens in prophase, prometaphase, metaphase, anaphase, and telophase, which we've talked about, but here's some things written for you. And you can see what it actually looks like. So you can compare it back to the animal cell pictures. So like, for instance, going back here, like prophase, we said looks like this, where the DNA is starting to condense, but you still have an intact nucleus. Here the nucleus begins to break down in prometaphase. So if we look here, prophase, here's the nucleus is still intact. You can see kind of dark spots, the DNA is starting to condense. But you can see it better in prometaphase. So the nucleus is starting to break down and you can start to see the chromosomes condense. In metaphase, they line up down the center of the cell. So in the computer-generated picture, that looks like this. In the real picture, that looks like this. Anaphase in the computer-generated picture, their chromatids are separating to opposite sides of the cell. That's what we see here. And then here, nucleus is starting to reform in telophase. You're starting to get the formation of that cell plate. So nucleus being reformed, And then this is the plant cell, so that's going to be a little bit different than the animal cell picture. The animal cell picture is the cleavage furrow. So I liked this because it gives you a real picture as far as what's going on and what it actually looks like inside the cell. And that is mitosis, basically. I do want to spend just a minute here and talk about how bacteria are a little bit different. Bacteria do not divide by mitosis. They divide by something called binary fission. And so this is, and that's because if you think about bacteria, what is true about bacteria? They don't have a nucleus. So prokaryotes, they have no nucleus. And anybody remember, do you guys remember how many chromosomes do they have? They have one circular chromosome. We have a nucleus, and we have 46 linear pieces of DNA or chromosomes. So it's different. So they don't have to do a lot of the same organization that we do in getting our cells to divide. So your prokaryotes, bacteria and archaea, the two domains, these guys are domains, reproduced by a type of cell division called binary fission. In binary fission, the chromosomes replicate and the two daughter chromosomes actively move apart. And this is a lot simpler than what has to happen in ours. All right, so here is one big circular chromosome. Remember, if you were to take it out and unravel it, the chromosome would be much bigger, circle would be much bigger than the actual bacteria. And so here's the origin of replication. So replication, what's going to happen? The DNA replicates here. I'm going to put start DNA rep. So it starts to replicate. So here's the copy, all right? And it begins to make a copy of its DNA. And so it begins to, replication continues. And so it eventually ends up with two copies of the DNA. So here's one, whoops, off the thing here, sorry, continues to copy, makes more copies. Notice here it's also elongating, like from here to here, the bacteria is getting longer. That's kind of some similarity between binary fission and mitosis. And eventually you have two copies of DNA. Here's one copy, and here's the exact copy. And the cell pinches in two, and you get two daughter cells, and these guys are exactly identical. All right, and that's basically what happens during bacteria. It's pretty simple. And the last part of this is to kind of link what happens in bacteria to what happens in eukaryotic cells like this, the evolution of mitosis. Since prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. And fossil record shows prokaryotes were around before eukaryotes. Certain protists, these are unicellular eukaryotic cells. Certain protists exhibit types of cell division that seem intermediate between binary fission and mitosis. So if we look here, binary fission let's make a copy. The copy goes on either side, cell divides in half, and there's no nucleus, nothing else to contend with. Unlike down here, which is eukaryotic cells like us, most eukaryotic cells have to get rid of the nucleus, and then you have your centrosomes with the spindles that attach and separate your copies to opposite sides of the cell so the cell can split in half. So those are the two extremes that we talked about, prokaryotes and eukaryotes like this. Now, these are the examples of intermediates, these dinoflagellates. Notice here that the nucleus stays intact here, and microtubules extend through the nucleus from either side. There's not a centrosome, but they extend, and the chromosomes make copies and separate to opposite sides of the nucleus, which then divide and so on. So it's kind of like not quite like this and not quite like this, but it's kind of intermediate. Same kind of thing with the diatoms. So these guys actually have kinetochore microtubules. See, now these guys, the chromosomes were attached to the wall of the nucleus and spread apart, and the nucleus spreads and divides in half. These guys now have kinetochore microtubules. The nucleus isn't disintegrate or breaking down like it does here. So again, an intermediate here, and all the nucleus divides and so on. But it's similar, has some similarities with these non-kinetochore and kinetochore microtubules to mitosis, but also some similarities to binary fission. And so we have some intermediates there, which leads people to believe that that mitosis may have evolved originally from binary fission and has become specialized because of the parts inside of it. All right, and that is cell division in a nutshell. And so we'll talk more about this Monday when we get back. And then we'll look at, if we look at the third section, the cell cycle is regulated by molecular control systems. So we're going to get into, notice here, cytoplasmic signals. We're going to pull in, again, signaling and cell communication and that kind of thing. But I hope that you are having a great break and getting some sleep and some rest. The video is only 45 minutes, so and I will get the review sheet up soon. Have a great break. I'll see you next year.